Hey there, future scientists and biology lovers. Welcome back to the channel. Today we are dividing into one of the most fascinating processes in the world of cells, meiosis. Now, why you should care about the meiosis? Well, meiosis is why each of us biologically unique. It's the very reason siblings don't look like identical clones, like an identical clone. So it's the secret behind the genetic diversity, the foundation of the sexual reproduction, and the creator of the gametes. You know what are the gametes, right? So gametes, they are sperm cells, right? They are the sperm cells and they are the ovum in the females, right? Sperm and egg cells are called the gametes. So imagine this inside your body right now. Cells are splitting, swapping DNA, and carefully halving their chromosome number just so that new life can one day begin with the right genetic code. So if you have a right genetic code, if the chromosome number is carefully halving, so that means it's imagine. So imagine this is a very good, fascinating topic in the world of biology. So sounds magical, right? But it's all the same. And today we're going to break it all down in a way that it's super clear, it should be visual, it should be unforgettable. So grab your notes, let's walk through the wonder of the meiosis step by step. So at the end of the meiosis, we will have four cells, all of them haploid in number, with a complete set of chromosomes. Okay, so meiosis one actually creates the genetic diversity. So before talking about this, let's talk about the stages of the genetic mixing. So first of all, as we talked in the mitosis phase divided into the prophase, metaphase, anaphase, and telophase. If you haven't watched our video about the mitosis, please, I recommend you to watch, okay? So prophase one, let's start from the prophase one. So I divide the meiosis into two. So meiosis one and meiosis two, there are two cycles, okay? So in the meiosis one, we have the prophase one, metaphase one, anaphase, telophase one, and cytokinesis one also. So prophase one, what's going to happen in the prophase one? Everything is happening, every magic is happening in the prophase one. Why? Prophase is where the magic begins, chromosomes start condensing, but the real show is called the crossing over. So what's crossing over? Crossing over means, first of all, in the prophase, what's happening? So homologous chromosomes are going to find each other. Here you can see the homologous chromosomes. This is one chromosome, this is the second chromosome, right? So this chromosome came from dad, this chromosome came from mom. They're going to find their partners. They're going to find their partners in the meiosis one, in the prophase one actually. So what's going to happen? They're going to do the synapses. They're going to do synapses. What's synapses? They're coming up together. These chromosomes are going to come up together. They're going to come closer. This process is called synapses when they join together. Okay, all right. So here, this is one chromosome. This chromosome is going to go near to the second chromosome, which is homologous chromosomes. They are homologous chromosomes, same in their sizes, same in their shapes, and also they are, but the difference is same in their genes, but the difference is in the alleles. They differ. For example, in one part of the chromosome, imagine we have the chromosome. In one part of the chromosome, if there is a gene, we have two chromosomes. They are the same genes contained in every locus of the chromosome, but they differ in their alleles. For example, imagine here that in the chromosome which came from that, there is a gene which encodes for the brown color of the eyes, color of the eyes, but here there's a blue color of the eyes which came from mom. They differ only in the alleles, okay? It's the homologous chromosomes, but they are same in their sizes, in their lengths, in their shape. They are all the same. Chromosomes are called homologous chromosomes, okay? I hope you understood the concept regarding the homologous chromosomes. So homologous chromosomes, came from the mom, came from the dad. They pair up together, and the parts of their DNA are swapped at the point called chiasmata. So I also said about the synapses. So when they come up together, when they came up together, you see these points. When they join, when the ends are joining together, site of the crossing over, the site of the crossing over occurs. This part is called the chiasma. All right. So also we can call the chiasmata as well. So this process known as genetic recombination is our first step toward the genetic diversity. So first of all, synapses happening, they are joining together. At the chiasma, they are exchanging their information. They are exchanging their information. This is the one chromosome, the homologous chromosome. This is another, right? They came up together very close. They are coming up together very close, right? At the chiasma, they are exchanging their information. This information is exchanging with this one, this is with another one. So they are exchanging their information at the site, which is called the chiasmata, okay, which is called chiasmata. At the end, we will have two chromosomes, and you can see the ends of the chromosomes are changing their parts. The red one came to the black one, the black part came from to the red one. So they exchanged their information. This is called the crossing over. Exchanging of the information occurs between the non-sister chromatids. And you know the sister chromatids. This is the first sister chromatid. This is another sister chromatid. This is another. This is another. So this sister chromatid is differing from this sister chromatid, right? So two non-sister chromatids are exchanging their information. This is called genetic recombination. This is called genetic recombination. They are exchanging, moms and dads in genes are exchanging with each other. That's why we are not identical. We are not the exact clones of our parents. That's why this is the first reason why we have the genetic diversity. All right? I hope you understood this. So, um, in the prophase one, a lot of processes look like to the prophase of the mitosis. But the difference, synapses, the chiasmata, the genetic recombination, crossing over, all of them, recombinant chromatids, they are all the unique processes which are unique in the prophase one. You have to know them. So what's going on here? Spindle fibers are going to form like in the prophase of mitosis. Please watch the mitosis process and actually the prophase one in order to understand this. I have explained the spindle fibers in the mitosis prophase. So please go and watch, and spindle fibers are going to form. The nuclear envelope breaks down in the prophase one also. I wrote everything as a note here. So nuclear envelope breaks down and the chromosomes are condensing in this part. What's the meaning? You see here the chromatids, right? Or I can say they are the DNAs. So what they are doing, they are condensing now. They're condensing, they're becoming visible in the prophase one. It's like a prophase of the mitosis as well. So centrioles move to the poles in this case. Centrioles are going to move. I explained the centrioles in the prophase of the mitosis, and the formation of the spindle fibers are happening. So this is the all you should know about the prophase one. You should all know. Please make notes regarding this. The genetic diversity number one is happening here in the prophase one because of the crossing over. They changing their information. Two chromosomes are changing their information. So what's going on in the metaphase one? Lining up randomly, right? This is the second genetic diversity. They are randomly lining up. Next up is metaphase one. Here, those homologous chromosome pairs line up in the middle of the cell. You see the homologous chromosomes. Homologous chromosomes are unique in the meiosis one. So you see the homologous chromosomes, right? They are very close to each other. These are the spindle fibers which are attached to the homologous chromosomes, all right? So what's going on here? But the key word is randomly. They are randomly. So I mean the homologous chromosomes pairing up together in the middle of the cell, but the key word is they are randomly. We don't know which one came from dad and which one came from mom. We don't know, right? We don't know which one is belongs to which organism. This random arrangement is called independent assortment. This random arrangement, the random arrangements of chromosomes We don't know. This chromosome is going to another pole. These chromosomes are going to another pole. We don't know which one came from dad, which one came from mom. This is called the independent assortment. This is the second type of the genetic diversity. This is the second genetic diversity. This identifies the second genetic identity. I hope you understood. The tetrads line up on the equator plate. Why we call them tetrads? What are the tetrads of the chromosomes? Tetrads of the chromatids, sorry. So we know the chromatids here. How many chromatids here? Two chromatids. This is two chromatid chromosome. Here we also have two chromatids, right? So if you sum up together, because in the metaphase they are together, if you sum up together, there will be four chromatids. We call them tetrads. So tetrads, they line up on the equatorial plate. This is the equatorial plate. These are the centrioles and the poles. So tetrads order in the random. They're randomly ordered now, the tetrads. So this is the independent assortment which gives us genetic diversity. So centromeres of the chromosomes attach to the spindles. So centromeres, there in the center we have the centromeres which connect to sister chromatids. Centromeres of the chromosome attached to the spindle. Spindle fibers are attached. So this is called tetrads of the chromosome, all right? So four of them is called tetrads. I hope you understood. Spindle fibers attached to the centromeres and tension builds here. So what's going on in the anaphase one? What's going on in the anaphase one? So anaphase is splitting the pairs. The pairs are splitting in the anaphase. So homologous chromosomes, which came from mom and one came from dad, we don't know which one belongs to which, separate into tetrads. All right. So sister chromatids remain together in this case. So you see the two sister chromatids here, two sister chromatids, they remaining together with centromeres. So centromeres are not splitting like anaphase of mitosis, they are still there, but homologous chromosomes are going to split from each other. All right. So spindle fibers, these are the spindle fibers, they're going to contract and pull the chromosomes toward the poles. They are pulling the chromosomes toward the poles. So the tension breaks here, the spindle fibers pull apart the chromosomes to the opposite poles of the cells. So you should note this, the sister chromatids stay together. All right, for the exam purposes you should know. Unlike in mitosis, the sister chromatids are staying together. This is important. We are only separating the pairs for now. Each daughter cell is still has duplicated chromosomes. All right, each daughter cells, these are the cells. So each daughter cell has duplicated chromosomes. You see, this one is daughter cell, this one is another daughter cell. They have the duplicated chromosomes now. They're duplicated, okay? So you should know about this. So what's happening in the telophase one? In the telophase, now the chromosomes arrive at the poles. The chromosomes are at the two poles now. Nuclear membrane may reform. Nuclear membrane is reforming here in this process. And each new nucleus contains only half of the original number of the chromosomes. They containing the half of the chromosomes. At the beginning, we had in one cell 6 chromosomes. In one cell, we had 6 chromosomes at the beginning. Now they are in one cell is containing 3 chromosomes, another cell is containing 3 chromosomes, all right? So they are haploid in number. 2 cells haploid in number. You should know this. That means that these cells now haploid, but they're not done dividing yet. This is just telophase 1. So what's happening in the cytokinesis one? In the cytokinesis one, finally the cell divides into 2 genetically different haploid cells. Please watch the cytokinesis and the mitosis as well. I explained everything clear. Each has half number of the chromosomes now at the cytokinesis one, but each chromosome still has 2 sister chromatids. You see, still 2 sister chromatids together. And now, without even pausing to rest, these 2 cells move to the meiosis. These 2 cells are going to move to the meiosis II. All right. So what's going on in the meiosis II? Let's break them down. So meiosis II, the mitotic-like speed. So they look like very to the mitosis. So before watching the meiosis II, please watch the video about the mitosis. Okay. So prophase II. This is the preparation round. So in the prophase 2, the nuclear envelope dissolve again. The nuclear envelope, we started here, there is no nuclear envelope, all right? Then they dissolve it again. New spindle fibers are going to form here. I'm not going to explain it briefly because I have explained during the mitosis. Please watch it, make notes. This time there is no crossing over, no crossing over here, all right? You have to know, no crossing over in the prophase 2, crossing over only in the prophase 1. So this is time, there is no crossing over, we are just getting ready to separate sister chromatids. We're going to now separate the sister chromatids, okay? Spindle fibers are going to form, degradation of the nuclear membrane, nuclear envelope, nucleolus are going to happen. The nucleolus is also going to disappear in the prophase 2. So what's happening in the metaphase 2? In the metaphase 2, each chromosome lines up at the equator again, each chromosome lining up in the middle of the cell, okay? Spindle fibers attached to the chromatids. Spindle fibers are going to attach to the chromatids. So after that, ready to pull them apart. So this step is all about precision. So in the anaphase 2, now we split the chromatids, okay? In the anaphase 2, this is like an anaphase of mitosis. Chromosomes divided the centromeres into sister chromatids, okay? So we are going to split in the metaphase, all the chromosomes will be in the center of the cell, right? So what's going to happen in the anaphase? In the metaphase also, the spindle fibers are attaching to the centromeres. So what's going on in the anaphase 2? They're going to pull, these spindle fibers are going to contract. They're going to contract and they're going to pull each sister chromatids. So we are dividing the sister chromatids. We are breaking the centromeres and we are splitting the sister chromatids into two. It's like the anaphase of the mitosis. All right. Spindle fibers are contracting and finally the sister chromatids are pulling apart to the opposite ends of the cell. This ensures that each gamete ends up with a single copy of the chromosome. Okay, each cell are getting the single copy of the chromosome. Good. So in the telophase 2, this is the finish line. Okay, this is the finish line. Chromatids reach to the poles again, and the new haploid nuclei is going to form. Okay, nuclear membrane is going to form again. Now we have 4 separate areas with just the right amount of the DNA. So at the end we have four haploid nuclei is going to form. 4 haploid nuclei. This is just this. From this one we will have one nucleus. From this one we will have another. Do you remember at the end of the telophase? How many cells we made? I only showed you this one. I only here illustrated for this one, for only one cell, and we have the another cell as well. From another cell we will have the same process. These 2 cells are undergoing the same process. That's why at the end we will have 4. This is from one cell, okay? This is from one cell, but we have the another cell as well. This is the first cell. We have another cell here exactly like that, okay? Let me copy this one as well. So I'm going to copy it, and okay, we will have 2 cells at the beginning, and at the end what we are going to have at the end we are having we call them, this is this one is come in the anaphase. This one came from this cell, these 2 ones, and also from this cell we will also have 2 ones, right? Sorry, I have to delete it, sorry. And I'm making 2 cells. I'm going to copy from this cell also. So at the end we will have 4 nuclei or 4 cells. Each of them, each of them are doing the cytokinesis at the end. This is called cytokinesis 2. Results in the 4 non-identical haploid daughter cells, and each one is going to contain haploid number of the individual chromosomes. We call them individual chromosomes now. I hope you understood this process. In the cytokinesis 2, 4 unique cells are producing. with one last division of the cytoplasm, now we have four non-identical haploid cells. These are, we call the gametes, okay? Each one is called the gamete because the meiosis only divides the gametes. Only they form the sperm or egg cells, each with unique combination of the genetic material ready to prepare in fertilization. And that, friends, is how life gets in variety. I hope you understood. Before we wrap up, let's compare the mitosis and the meiosis, okay? Let's recap. So, as we know, the mitosis produces two identical cells. In the mitosis, we will get two identical cells, but meiosis produced four non-identical cells at the end. We call them gametes, okay? So mitosis keeps the chromosome number the same. The mitosis is keeping the chromosome number the same, but the meiosis is going to halve it. So what about crossing over? Crossing over is only happening in the meiosis I, okay? So crossing over is only happening in the meiosis I. And the pairing of the homologous chromosome is also happening in the prophase I. Okay, so separation of the homologous chromosomes are happening only in the meiosis I, in anaphase. Crossing over happening in the prophase I. This is anaphase I, this is prophase I, and the pairing of the homologous chromosomes are also prophase I. Okay, centromere splitting is happening only in the mitosis and the meiosis II because the meiosis II and mitosis, they are exactly the same. Separation of the sister chromatids also happening in the anaphase II or in the mitosis. They are the same. You have to know. So I hope you understood everything. Number of the rounds of the DNA synthesis in both of them, we have only one interphase, one S phase for all of them. That's why it's happening once. Number of the cell divisions in the meiosis II and mitosis I. Number of the total cells. At the end of the mitosis, we get two cells which are identical to the parent cell. At the end of the meiosis, we get four cells which are not identical to the parent cell. Okay. So chromosome complement of the daughter cells compared to the parent cell, they are the same. They are the half number. They are haploid in the meiosis. This is diploid in the mitosis. So you have to know it. So I hope you understood everything. To sum up, meiosis is a brilliant, complex dance of the division that creates the diversity. That it's creating the diversity of the life, and it's how we go from one diploid cell to four unique haploid cells, each a potential beginning of the new human history. If you found this helpful, leave a like, subscription for more amazing biology content, and don't forget to check out the comments for your quiz challenge. See you next time.